presentation. So I was saying that in addition to her outstanding research accomplishments, Dr. Martin is really a dedicated mentor, and she guides scientists at all career stages, from undergraduate trainees to faculty, and I can personally attest to her exceptional mentorship, having benefited from her invaluable feedback when navigating challenges, writing grants, and all that. Her international leadership is exemplified by her recent role as co-chair of the 2025 Physiology, Biology, and Pathology of Phosphate, a Gordon Research Conference. That really is a very unique event with international leaders in the fields. So with that, I will close, and please join me in welcoming Dr. Martin.
Wow. Thank you so much, Penelope. That was a very kind introduction. And thank you for having me today. So just to pause on the title a little bit, I think, the last time I presented renal grand rounds was many years ago, and a lot obviously has happened and changed since, but we can probably all agree that one thing that remained constant throughout the years is probably that question in the title: how do we treat bone disease in CKD? So we kind of hang tight to that question. When you approach colleagues in the field and you ask that question, it's kind of an embarrassing question to ask. And for me, it became a little bit of a game of truth and dare. And so you get many different answers, but the most frequent one you would get is, well, we don't really know how. It's complicated. And so it is complicated. It's actually, in a way, our embarrassing truth. So obviously today, I don't intend to solve that big question, but what I would like to do is review with you some potential pathogenic mechanisms of CKD-associated osteoporosis, talk about bone disorders in altered phosphate homeostasis. I'll present you some work that we did in the lab that support direct effects of FGF23 on osteoblasts. I'll come back to that and discuss the relevance of this in CKD. So why is it so complicated? So this is kind of a simplified summary of CKD and BD. And as all of you know, as kidney function declines, patients with CKD will experience a combination of these secondary complications that are all more or less interconnected, and they heavily contribute to mortality. And so today, I'll focus on altered bone and mineral metabolism, and we'll leave the rest for another day. But so CKD is associated with a significant risk of fracture. And this is amplified with CKD progression and with age. And this is what you see on this graph, compared to the general population, which is plotted in the black line, and then you have the different CKD stages in colors up to hemodialysis patients here. So it's very high. Even after kidney transplant, we continue to see a decline in bone mineral density, and that was shown here in this study, mainly in the peripheral skeleton here. So currently, there is not really a therapy that is specifically designed to address impaired bone quality in CKD. And the problem that we have is that we don't really know what's going on. We don't fully understand the mechanisms that drive the bone disease in CKD. So to understand bone a little bit better, I put this slide up. Bone is an endocrine organ, and it's constantly remodeled during balanced cycles of bone resorption that is handled by the osteoclast cells, pointer here, and bone formation and mineralization handled by osteoblasts and osteocytes. And as the osteoblasts differentiate, some of them will become osteocytes. they'll populate the newly formed bone matrix and support the mineralization process by secreting various factors, including, for example, DMP1. And during the mineralization process, calcium and phosphate get incorporated into the newly formed bone matrix in the form of hydroxyapatite crystals. And the disruption of those cycles of bone remodeling and mineralization is often what results in bone disease. So, for instance, in postmenopausal osteoporosis, we are looking at too much bone resorption in the face of too little bone formation. So we lose bone. In CKD, it's also a little bit the same. Bone remodeling is altered, but that's not the full picture. There's also impaired mineralization, and there's accumulating evidence that suggests that this cell, the osteocyte cell, might be at the center of the development of the bone disease in CKD. So these are electron microscopy picture of cortical bone osteocytes, and they show an altered morphology of the osteocyte cell in both human and mice with CKD compared to healthy controls, regardless of the stage of CKD progression and of the age. So really, it doesn't matter if you're an older person with early CKD or a young person with advanced CKD, the osteocyte will show as a more circular cell and have less cytoplasmic dendrites, so to speak, compared to healthy elongated cells that it's supposed to be. So there's a few studies, including these, that were performed in the pediatric CKD population that indicated that osteoblast maturation might be impaired in CKD, and that could be due to significant epigenetic change and modifications in the osteoblast cell. More recently, the David lab from our division at Northwestern published that patients with CKD and who have either low or high bone remodeling, so LR or HR, showed so differentially regulated genes. So this is RNA sequencing of the bone in bone biopsies from patients. So these genes were differentially regulated and contributed to impaired osteogenesis, metabolism, and to cell death. And among these genes, the transcription factor HNF4 alpha 2, to be precise, was discovered to be expressed also in bone. It was heavily repressed in CKD, and rescuing its expression in mice with CKD corrected the development of the bone disease. And together that proposed HNF4 alpha as a new key regulator of osteoblast metabolism that's involved in CKD also. And a few weeks ago, there was, and kind of in line with this analysis, there was this paper that came out in KI that showed that mitochondrial function is altered in osteocytes in response to uremic toxins. And this likely impact mineralization and cell death, although that was not directly really shown in that paper. And to me, I mean, also one of the big questions that remains is what are the proximal factors that regulate this process in osteocytes? In this study, it might have been HNF4 alpha, we don't know. It could also have been different factors. So at this point, this is still an open question. And then perhaps one of the most simple and I want to say exciting observation that was made on osteoblasts in CKD is the fact that they retain mineralization defects when they're isolated and cultured outside of the systemic environment, pointing to possible local modification of the osteoblast cell and that persist, outside of or in the dish. And so this here was performed by the group of a collaborator, where they isolated cells from bone biopsies that were taken from patients with CKD or healthy controls. And then they cultured those in osteoblast differentiation medium and performed an alizarin red assay that quantifies bone mineralization. And so what they showed is that mineralization is reduced or delayed in CKD osteoblasts. And we were able to show the same thing in my lab after isolating osteoblasts from mice with CKD or healthy mice, so the wild types or the COL4A3 knockout, which is a model of progressive CKD. In addition to the mineralization staining, we did an alkaline phosphatase staining that quantifies osteoblast activity, and we showed that both osteoblast activity and mineralization were decreased in CKD osteoblasts compared to controls. So another interesting thing that we found here was that one of the factors that was persistently highly expressed in these cultures, even after 3 weeks of culture, was FGF23 expression. So FGF23 was highly, highly expressed in culture. So I'm going to talk about FGF23 mostly for the rest of the talk. So just before we get back to those cells, normally the osteocyte is the cell that mainly produces and secretes FGF23. Together with PTH and vitamin D, FGF23 is a hormone that's going to coordinate the regulation of mineral metabolism, and they do so by balancing intestinal absorption with bone mineralization and renal excretion. And that together maintains a stable calcium phosphate balance. And in the kidney, the established function of FGF23 is to inhibit phosphate reabsorption and inhibit calcitriol production. For phosphate, how it works is that FGF23 will bind to FGF receptor and Klotho complexes in the proximal tubule. The downstream signal will inhibit the expression and internalization of the sodium phosphate co-transporters here and here. And as a result, FGF23 induces increased urinary phosphate excretion. Okay, here in red, increased FGF23 is the earliest alteration in mineral metabolism that occurs in CKD. And levels continue to rise, and they exponentially rise as patients reach end-stage kidney disease. After FGF23 rises, we see a decline in vitamin D levels, that's your blue line here, and that contributes to the development of hyperparathyroidism, which is the green line. And all these modifications will contribute to maintain serum calcium stable up to a point when kidney is no longer able to sustain enough phosphate excretion that, and then serum phosphate levels start to increase as well. So to get back to our bone cells, then we wondered whether all this FGF23 could contribute directly to the bone disease in CKD. And there were several, or just a few actually, sporadic studies that hinted to direct effects of FGF23 in bone in the past. Most of those were in non-disease settings, and they showed a lot of in vitro work. And the most recent one was this one at the bottom right. And in this study, it was performed already 8 years ago. So in this study, what they did is they took a rat model of CKD and they challenged it with dietary phosphate supplementation, so a high phosphate diet, to increase FGF23 levels. But what that did is that it also increased PTH levels. That's what you would expect. And to try to disentangle the effects of FGF23 from the effects of PTH, they brought this in vitro. They did some work separating FGF23 effects on bone cells from PTH effects. But, so the direct effects of FGF23 on bone in vivo were not shown, despite the title. But in their defense, it's actually complicated. It's complicated to do. So to try to uncomplicate this a little bit and to address our question whether FGF23 has effects on bone directly, we decided that we could take out kidney disease from the equation using non-CKD models of FGF23 excess. And these include models of hereditary hypophosphatemia. So in CKD, what triggers the initial increase in FGF23 production is not so clear. Likely, it's a combination of factors or events that are initiated by the kidney injury. In hereditary hypophosphatemia, it's more simpler because, in a way, because monogenic mutations in the FGF23 gene itself or in upstream regulators of FGF23 in the bone will trigger a primary increase in FGF23 production in the bone in absence of kidney injury. And so in this case, FGF23-induced renal phosphate excretion will result in severe hypophosphatemia, so low serum phosphate, which is a main difference with CKD, where serum phosphate levels are high. Okay, also contrary to CKD, hereditary hypophosphatemic rickets are rare diseases. In hereditary hypophosphatemia, the primary FGF23 excess and hypophosphatemia are associated with impaired bone growth, impaired mineralization, and rickets, which is the bone deformation that you see here on those pictures. The symptoms often appear during infancy, and then they possibly become worse in ARHR1, which are the DMP1 mutants, compared to XLH over time. We don't really know why that's the case, but it is what it is. And so these syndromes have been extensively studied using FEX and DMP1 mutant mice that really recapitulates what you see in patients with XLH and ARHR1 respectively. And so previous studies have shown that these two models, the HYPE and the DMP1 knockout, they mostly phenotypically overlap. So they show an increase in circulating FGF23 levels, about at the same level, and they all also show hypophosphatemia. So it's generally accepted that hypophosphatemia, so low serum phosphate, is the main contributor of what happens in the bone in these models, so low mineralization and impaired bone growth. But the work I'm going to show you next kind of brought us to a first interesting observation for the purpose of this talk, is that it's actually not all about phosphate. And by the way, don't repeat I said that to the people who attended the meeting I just organized in February in Italy, which was all about phosphate. So why am I saying it's not all about phosphate? So before I go on over this slide, I should say phosphate is required for mineralization, but more phosphate does not necessarily equal more mineralization. So there's a subtle difference there. And I think this study is actually the best illustration of that concept. And so some of you might know the conventional therapy for XLH and ARHR consists of phosphate repletion in the form of oral phosphate supplement and calcitrol and vitamin D analog therapy. However, oral phosphate supplementation has been largely ineffective at correcting bone defects and hypophosphatemia in XLH, mostly because oral phosphate will further increase FGF23 And that, as a result, further increases urinary phosphate excretion. So basically what happens is that whatever phosphate comes in immediately comes out. And so that works in human and mice. And so we, in the lab, took DMP1 knockout mice with ARHR1, and we did basically the same thing. So these mice were fed, we did a detailed analysis of mineral metabolism and bone metabolism. And so DMP1 knockout mice are fed for 6 weeks on a control or a high phosphate diet from 6 weeks of age. As I was explaining, so your DMP1 knockout on control diet is the red group here, and then on high phosphate is brown. And so as I was explaining, high phosphate further increased FGF23 excess. So serum FGF23 levels go up. We usually measure FGF23 in the serum using 2 different assays in the lab. We don't have to go in details on that today, but that's what you see here. And then high phosphate also increased serum PTH levels here. Together, these further increased urinary phosphate excretion, so fractional excretion of phosphate is further up, and that contributed to maintaining serum phosphate levels low. So we have persistent hypophosphatemia here. With that, looking at the bone, the high phosphate diet did not rescue any of the bone parameters or bone growth. We still have a significant growth defect. Looking at micro-CT parameters, we didn't see any improvement, and actually we saw a worsening of the trabecular and cortical bone mineral density, and a further expansion of the cortical bone that you can see here. So the bottom line is that whatever phosphate these mice ingest, they excrete it right away, the bone doesn't improve, and actually it's even a little bit harmful. So to move on from that, in recent years, FDA has approved a new drug that's called, it's an anti-FGF23 antibody called burosumab, and it blocks FGF23 function. How it works is it's going to bind to FGF23, prevent it from binding to the receptor clotho complexes in the kidney, and prevent its phosphaturic function. And so burosumab has shown superior efficacy compared to conventional therapy in terms of rickets severity score. That was improved. So the conventional is pink, burosumab is blue, and also improving serum phosphate levels that came back in the normal range following burosumab treatment. At this time, it's not so clear what happens to bone remodeling. So I guess future studies will tell us a bit more about that. There's quite an improvement on the bone, but we don't have bone biopsies to tell us about bone resorption, bone formation, and so on. Burosumab, however, is not yet approved for the treatment of ARHR1. ARHR1 is super rare, so there was only one case report that was published 2 years ago that reported treatment on 2 brothers with DMP1 mutations, and this study was promising because it showed an improvement of serum phosphate levels after initiation of the treatment. So that's blocking FGF23 function. Before burosumab was approved, this next study actually showed interesting preclinical results, repleting serum phosphate levels, not by blocking FGF23 function, but by reducing FGF23 excess, reducing FGF23 levels. And the way they did that, so this was done by the group of Ken White in Indiana, and the way they did that is by specifically deleting, in a mouse model of XLH, specifically deleting FGF23 in osteoblasts using a collagen type 1A1 CRE that targets osteoblasts from the early stage of differentiation. So they targeted a large population of osteoblasts. And so doing that, they were able to rescue FGF23 excess, which, so you see circulating FGF23 levels are actually a little bit below normal, but still detectable. They're not completely wiped out. And that fully corrected hypophosphatemia. So these are hype mice with FGF23 deletion at the center here, and corrected hyperparathyroidism. So the hype mice with FGF23 deletion looked comparable in appearance to the wild-type mice. They no longer showed any growth defects, and you see it here also with the femur length and the tail length that was quantified. And the shape of the bone here was also rescued compared to the initial ricketic phenotype that you see here. Although they had like a little bit of something funky going on with the trabecular bone, that was not quite explained. We didn't really know what was happening there. But that study really demonstrated the role of FGF23 excess and hypophosphatemia in the pathogenesis of XLH at the time. So you fast forward 7 years later, and here at Northwestern, we took a similar approach to reduce FGF23 levels in the DMP1 knockout animal this time, DMP1 mutant. And so what we did differently is that instead of targeting a big pool of osteoblasts, we targeted only mature osteoblasts and osteocytes using a DMP1 CRE. And so what happens doing this is that we did correct FGF23 excess, although not completely. Serum FGF23 levels were corrected by about 80%. PTH levels were corrected by about 50%. Together, that reduced urinary phosphate excretion, and that correction was enough to fully correct hypophosphatemia. So we brought serum phosphate levels back to normal. So we were pretty happy about that, and then we said, okay, phosphate is completely corrected. We can look at the bone, see what happens. And this, that's calcium stayed normal. And then we looked at bone. So that's what happened. So the lowering FGF23 levels actually did not fully complete, and rescuing serum phosphate levels did not fully correct the bone phenotype. We still had a little bit of growth defects, and the shape of the bone also was not totally rescued, right? So we still have residual rickets here. The cortical bone porosity, which is increased in the DMP1 knockout, was partially rescued only, and you see it here as well. The trabecular bone volume, cortical bone thickness were partially rescued. Osteocyte morphology was partially rescued. Growth plate, which you see here, was rescued. However, the the mineralization was not rescued. So we still had impaired mineralization. So despite the full correction of hypophosphatemia, we did not completely rescue the bone phenotype and the bone mineralization, which is why it's not all about phosphate. And we had to look for phosphate-independent factors that were responsible for the residual phenotype here. So it also brought us to a second observation at that time, is that bone can actually be the center of attention. And I challenge you to find a bone on the cover of a non-specialty journal. This is kind of a once-in-a-lifetime thing, a little bit like a solar eclipse. And so we were very happy because within a 5-month time frame, well, we saw both. So we beat the odds. And more importantly, the reason why this study made the cover of the journal is because we demonstrated that in this model, FGF23 actually directly inhibits the differentiation of osteoprogenitors. And I'm going to show you how we did this. We brought everything in vitro, and we took these 4 groups. We isolated bone marrow stromal cells from them, cultured them for 3 weeks in osteoblast differentiation medium, and then at the end of those 3 weeks, we performed bulk and single-cell RNA sequencing. And so, using established markers of osteoblast differentiation, by single-cell analysis, we were able to identify 4 very nice clusters of osteoblasts from osteoprogenitors all the way to osteocytes. And this is also shown here in the pseudotime analysis, with purple being the least differentiated cells, osteoprogenitors, and then moving to the yellow, which are the most mature cells, the osteocytes. And so then we segregate this by group, and what we observed is a higher amount of osteoprogenitor cells in the DMP1 knockout group, and a lower amount of osteocyte cells, indicating an impaired differentiation of the osteoprogenitors in DMP1 knockouts. And so that was rescued with the osteoblast-specific deletion of FGF23, suggesting that FGF23 might have a role in that impaired differentiation. And so the next thing we did with that is to identify genes that were modified in DMP1 knockout, but no longer modified when we deleted FGF23. So we focused on that part here. We did this by bulk RNA sequencing, and also in each separate cluster of osteoblasts that we identified by single-cell RNA sequencing analysis. And we found that osteoprogenitors showed the highest number of modified genes. And then you run a pathway analysis on this, and what we found, consistent with an effect of FGF23 in the DMP1 knockout osteoblasts, we found that FGF receptor signaling, ERK, PI3 kinase, and NFAT signalings, which are downstream of the receptor, are all modified in the DMP1 knockout. Osteogenesis and cell morphology were also at the top of that list. So that was by bulk in the entire pool of cells. And then by single cell, we were able to find most of these pathways also, but only in the osteoprogenitor cluster and not in the more mature osteoblast cluster, suggesting that FGF23 targets osteoprogenitor cells in ARHR1. We next demonstrated the direct effects of FGF23 on osteoblast differentiation by treating osteoblast cell lines with recombinant FGF23 for a short time, and we showed direct inhibition of the expression of osteoblast differentiation markers. SP7, which is called also Osterix, RUNX2, and osteocalcin here. And so that was in an acute study. Long term, we evaluated osteoblast activity and mineralization in our 4 groups. After we co-cultured them with cells of the same genotype, which we call the isogenic control, or with cells that overexpressed FGF23 or that overexpressed DMP1. And so what we found was that in DMP1 knockout, we had a reduced osteoblast activity across the board. That was rescued by the deletion of FGF23, which is consistent with an effect of FGF23 inhibiting osteoblast differentiation. And then regardless of DMP1 expression, when you co-culture these cells with FGF23 overexpressing cells, all of them show reduced osteoblast activity. And so in addition to that, we found an interesting effect of DMP1 on mineralization that was not rescued by the deletion of FGF23, indicating it's a specific effect to DMP1. And that was only rescued when we co-cultured these cells with DMP1 overexpressing cells. So, consistent with this, by sequencing, we looked at genes that were modified in DMP1 knockout, but actually that remained modified after we deleted FGF23. And so we found all these different pathways that were related to these differentially regulated genes, including HNF4 alpha. And in single cell, we also found that these were represented in the osteocyte cluster, but not in the early osteoblast cluster. So unlike FGF23, this suggests that DMP1 would tend to target more mature cells, and including osteocytes. Okay, so next, in the time I have left, I want to bring this back to CKD. And so in the lab, I was telling you about this model earlier. The call4A3 knockout is a progressive CKD model, genetically induced, so you don't have to do anything, no surgery, no diet. And to give you a little bit of an overview of what's going on in these, so BUN levels start to increase at 12 weeks of age in this cohort, and then they continue to go up afterwards. We have FGF23 that starts to go up also at 12 weeks and exponentially rises after that. PTH levels, here, increase at 16 weeks, continue to go up, and then finally serum phosphate levels are increased at 20 weeks and go up. And so the bone phenotype of these mice, looking at parameters of cortical and trabecular bone microarchitecture, we see bone loss starting at 12 weeks that worsens after. And pretty much all markers of osteoblast differentiation, their expression is decreased in this model compared to controls. So we have a nice model where we see pretty much all the alterations of bone and mineral metabolism that we want to be able to study this. What we did next is challenge those with high phosphate diet. And so consistent with the previous models I showed you, even in rickets, or with the study that was done 8 years ago in the rat model of CKD, when we feed CKD mice, either a control or a high phosphate diet, this is what we see. So you have your FGF23 excess and hyperparathyroidism here. On high phosphate, you accentuate this a lot. So these mice were fed a high phosphate for about 20 weeks, so long, long time. And then looking at the bone, so that's consistent with the study that was performed in rats. And then when we look at the bone loss that we see in the CKD mice fed the control diet is also accentuated on high phosphate. By bone sequencing analysis, the pathways that are modified in the CKD mice fed the control diet were further modified on high phosphate diet. So we had the same issue, basically. How do we disentangle the effects of FGF23 from the effects of PTH? So we were a little bit stuck, but we were lucky because we started to study another model, which is a model of AKI to CKD transition that is surgically induced. So what happens is healthy mice undergo bilateral ischemia reperfusion injury, surgery at 12 weeks of age. Usually it's used as a model of AKI, and they're studied shortly after surgery. In our case, we wanted to look at the bone and mineral metabolism, and so we kept them for many, many weeks after that. And we studied 2 sets of mice, one at 20 weeks of age, so 8 weeks post-surgery, and one at 31 weeks. And so what happens is post-surgery, you see an increase in BUN levels, and BUN plateaus afterwards, confirming kidney injury and kidney disease. And then looking at FGF23 levels, what we see is an increase at both time points, an increase in serum FGF23 levels that worsens over time, despite BUN plateauing. So we were happy to see this. We looked at PTH, and interestingly enough, PTH did not change in the CKD mice compared to the sham control. It increases over time with age, but not between the two groups. And then phosphate levels, so over time with age you see a reduction in serum phosphate, and that did not happen in the CKD animals. That might have contributed also to increase FGF23 levels a little bit. So for us it was good because we had a model where FGF23 was high, but PTH was normal. And so we looked at the bone, and in this model what happens many weeks after surgery is that you do have bone loss, and that worsens over time. And that happens in the trabecular bone with these 2 parameters. Cortical bone, same thing, and then cortical bone porosity actually goes very, very high at 31 weeks of age. And to emphasize, this happened despite or in absence of hyperparathyroidism, despite stable BUN, and in association with increased FGF23. So at this point we were excited about this model. We said, well, that means perhaps FGF23 directly contributes to the bone disease in CKD. And that brought us to our third and last observation, is that sometimes perhaps we should pick a dare, and that might be less embarrassing than the truth. And so this thing about reducing FGF23 in CKD has created a little bit of controversy in the past because, I should say, partly because of one study that treated rats with CKD with burosumab, so the FGF23 blocking therapy. And as a result of this, so it was done with very high doses of the antibody. As a result of this, serum phosphate levels went really, really high. Mortality was enormous. And so after this, basically that was it. We didn't hear about it. And so for us, we said, well, we should take a less aggressive approach. And we tried 2 different things. So the first one, we kind of repeated what we did in the rickets model. We specifically deleted FGF23 in osteoblasts from the Col4A3 knockout mice with CKD. And in here, we targeted a large pool of osteoblasts with an osteric scree. So you have your CKD mice in red, and then the mice with CKD and FGF23 deletion in green here. The deletion of FGF23 did not impact CKD progression. BUN is still high, but not higher. And then we have a nice partial rescue of serum FGF23 levels. So that was lowered. We were excited about this. As a result, serum phosphate levels went up, but PTH did not change. So that wasn't worsened. Okay. And then, depending on which parameter of bone microarchitecture you look at, what you see is a partial to a complete rescue of the bone phenotype. So here you see the bone loss in the CKD animals, and that's completely or partially rescued in the trabecular bone compartment and in the cortical bone compartment here. So that happened also despite persistent hyperparathyroidism and worsened hyperphosphatemia, which is an important point to make here. Okay, and then the second, I don't know if I have time, maybe I have time. The second approach we took was that we used DMP1 as a mean to lower FGF23 levels in CKD. So you remember in DMP1 knockout, FGF23 is very high. We think how that works is that DMP1 binds with to FEX at the osteocyte cell surface, and then through intermediary steps that are not so well known, that inhibits FGF23 transcription. When either or both are mutated or deleted, then FGF23 goes up. So we used, in CKD, FEX is expressed, but DMP1 expression is reduced throughout the CKD progression. So we crossed our CKD model, the COL4A3 knockout, with a mouse that genetically overexpresses DMP1 in bone, and that's what you see here. So in pink, you have mice with CKD that have very high expression of DMP1 in bone. And so what we found is pretty much exactly the same as the results we had with the FGF23 deletion in osteoblasts, meaning we reduced serum FGF23 levels by about 50%. As a result, we accentuated hyperphosphatemia, but serum PTH levels were unaffected, and kidney disease progression was also not affected. And in the bone, we had a full correction of the bone phenotype by lowering FGF23 levels and increasing DMP1, I guess. We did a little bit of further digging, and consistent with the rest of the study, we fully rescued the morphology of the osteocyte in here, and that was accompanied with reduced osteocyte apoptosis in the bone. And then in vitro, overexpressing DMP1 also rescued the mineralization defect that we see in the CKD group. Okay, and so that's my last slide before the conclusion. It's a little bit of a bonus, and that basically shows that in both, using both approaches, either deleting FGF23 in bone or overexpressing DMP1 to lower FGF23 production, that prolonged the life of the mice with CKD by about 10%, which is pretty significant. You might know that FGF23 has been previously shown to be independently associated with mortality and cardiac hypertrophy, and we have additional data in the DMP1 cohort in this study, and that was published, by the way, that showed that overexpression of DMP1 and lowering of FGF23 in that cohort improved the cardiac phenotype, so we think that's related to that. All right, and so I'm not going to say more on this. I think that's for another day. To conclude, we're pretty confident that altered osteoblast differentiation contributes to CKD-associated osteoporosis. Increased FGF23 production by the osteoblast directly inhibits the differentiation of osteoprogenitors. We've shown that. And importantly, lowering FGF23 in CKD, at least in mice, prevents bone loss, and that happens despite persistent hyperpara and hyperphosphatemia. And so with that, I would like to thank all my lab members, past and present. They've done a fantastic job at gathering all this data, running all these long diet studies and genetic deletions. Thank you guys very much for your hard work. I'd like to thank our collaborators and our sponsors. And I thank you for your attention, and I'll take questions. done.